Hi, I'm Stephen Nichtberger, President and CEO of Cabaletta Bio, and appreciate the opportunity to share our story with you today. So, Cabaletta exists to develop and launch the first curative, targeted cellular therapies for patients with autoimmune disease. And now the biology suggests that we could cure the diseases that we are targeting. But in order to win for patients and for our investors, all we need to do is be substantially better than the best available therapies. In order to do that, we're developing these highly specific CAA CAR products to treat B-cell mediated autoimmune diseases. We're only focused on diseases where there's an opportunity for biologic cure, where there's an opportunity for clinically deep and durable responses. We're leveraging clinically validated CAR T technology out of the University of Pennsylvania, where both the product design and our manufacturing process have taken advantage of the successes that they've seen with the development of Kymriah. We've applied our this framework to our lead program around the DSG3 CAR T product, and the name of the trial is the DesCART trial. It's currently underway, with the first patient having been dosed at the end of last year. And we expect in the next few months to be able to report out the first data from that trial. The acute safety data from the low-dose cohort will be followed by dose cohort data sequentially throughout the year. Our preclinical pipeline is led by the Musk Myasthenia Gravis CAR. This product is on track for an IND filing later this year. And behind that, we've got a portfolio of 5 additional products, some of which we have not yet identified, that we're actually very enthusiastically making progress with. Our lead program already has its first patent issued in the US, and this is substantial because the patent, unlike the CD19-directed CAR T space, covers all of the target antigen that needs to be on the CAR in order to address the disease. So in contrast to the CD19-directed CARs, where a single antibody fragment is covered, all of the possible parts of the antigen that is required in a CAR to address an autoimmune disease is covered in this first issued US patent. Additional patents are filed on the rest of our portfolio as progress is made. Our cash runway takes us at least into and through the third quarter of 2022. The last couple of quarters, we've spent about $10 million a quarter. We have $118 million in cash at the end of September 2020. Over the past year, we've had a pretty substantial path setting the foundation for the coming year. So we achieved fast track and orphan drug designation and have now started to see increasing enthusiasm and progress on clinical trial sites opening in order to get involved in this trial. And in addition, the patients are showing up at the sites looking to have a cell therapy administered that offers the promise of a potential cure. So the lead clinical program has now entered a phase in its development where we would expect to see accelerating enrollment in the trial and a pace on the trial that's faster than we've seen previously over the past few months. The Musk CAR T program is, as I said, on track for an IND filing in the second half of this year. And the path to that IND filing is very similar to the DSG3 CAR T path that we took previously in accordance with FDA's guidance. So let's just take a look at what our foundational platform is. So as I said, we're based on Kymriah and its development at the University of Pennsylvania. And so to create that therapy, you start with a patient's T cell, and you design in a signaling domain and a co-stimulatory domain. On top of that, you put a targeting domain. The targeting domain is comprised of antibody fragments that identify and attach to the CD19 antigen, which is found on normal B cells, as well as on leukemia and lymphoma cells that are cancerous. After successful therapy, the CAR T cell will have killed all of the B cells in a given patient, the healthy and the cancerous cells, resulting in no humoral-mediated immunity in that patient. Our scientific co-founders, one of whom is an inventor of Kymriah, having worked under Carl June over the course of his career, and the other is a B-cell-mediated autoimmune disease expert. And the two of them got together and said, hey, if the entire process, the signaling chain, is left intact, now that we know it is the basis for a safe and effective product in the treatment of cancer, is there a way that we can design the targeting domain so that it does not address or kill all B cells, but rather it addresses and kills only the B cells that are causing the autoimmune disease? And it turns out that if you simply keep everything the same, but change only the targeting domain from the antibody fragments shown in the cancer example, and you replace it with the antigen that's the subject of attack in an autoimmune disease, that antigen that is the subject of attack will be attacked by B cells that have on their surface copies of the antibody that they would want to produce. Those antibodies will bind to the targeting domain on our CAA CAR, and the B cell will be destroyed, just like cancer cells are destroyed, by the same signaling chain as is used with Kymriah. The healthy cells in our patients do not have the necessary antibody or B cell receptor on their surface, and they won't be recognized by our product, allowing for the normal healthy immune system to be maintained after successful therapy. So this biologically elegant approach has been applied in Pemphigus, both of its forms, and as I said, later this year, we expect to enter the clinic with our Myasthenia Gravis product as well. So let's dive deep into the lead clinical program. Why Pemphigus? Well, it turns out the DSG3 antibodies that are known to cause Pemphigus are both necessary and sufficient to cause the disease. That means that if you get rid of those antibodies, you don't have the disease. And so it's a nice linkage between the biology and the clinical outcomes. We can have expectations that if we can get rid of those antibodies and keep them gone, we can end up getting a durable curative response. So in order to help you think through this as we have, let's walk through these five different steps. So the first is the serum anti-DSG3 antibodies are 98 to 100% sensitive and specific for the disease. If you have the antibodies, you have the disease. If you don't have the antibodies, you don't have the disease. Depletion of the B cells by rituximab or by plasmapheresis reduction of the antibodies leads to a transient improvement in clinical disease. So we know that the B cell modulation would result in clinical outcomes that are improved. Incomplete B cell depletion by rituximab leads to recurrences, very reliably so, recurrences of the disease. And the recurrences are caused by clones that were previously present. Now, this has been studied longitudinally, and there are no cases reported where a novel clone appears which was not already present prior to treatment. So we know that B cell depletion can work, but incomplete B cell depletion reliably, unfortunately, is associated with recurrence of disease. The B cell repertoire and the antigenic epitopes that are triggering an antibody response are very well understood. And this allows us to design our targeting domain to incorporate all of the various epitopes that would be triggering the antibody response that causes the disease. And finally, the DSG3 CAR has published animal model data in Science. So with that as a foundation, Pemphigus vulgaris is really the optimal disease for us to prove the entire platform of CAR T technology addressing B-cell mediated autoimmune diseases. There are existing therapies, and they tend to be broad immunosuppressive therapies that are associated with reliable recurrences as well as safety risks. So the two forms of pemphigus are shown here, one mucosal in the mouth, the eyelids, and other orifices, and the mucocutaneous form, which is in all of those locations, plus diffusely across the skin. Our lead program is targeting the anti-DSG3 form. Current treatment is enhanced greatly a couple of years ago by the approval of rituximab. And rituximab plus about 2.8 grams of steroid per year will result in a response in about 40% of patients achieving a 4-month period with no lesions or medicines during the first year of therapy. There's associated with this somewhere between a 5 and a 9% annual risk of severe infection. Real-world data suggests that you can get a transient remission of disease with rituximab in about 70% of patients. Unfortunately, of those who respond, a third will have a recurrence during the first year, and another third will have a relapse during the second year. So at the end of the day, there have been important advances in care, but patients and their physicians are clamoring for better therapies for this disease. To give you a sense of how we design our products so that any patient with the disease would be appropriately treated with the DSG3 CAR T product, let me take you through this slide very briefly. The DSG3 protein is shown on the top left, and it contains 5 extracellular domains. Above those domains, the percentage of patients who have antibodies to each of the domains. So each patient has multiple antibodies that are going to attack the DSG3 protein causing disease. EC5 is known not to cause human disease. We designed into our product only EC1 through 4 as a result. When our DSG3 CAR T product is formed, shown on the right of the slide, we identify with each of these domains the antibody and the portfolio of epitopes that would be addressed by that part of the targeting domain. And as a result, you can see that all known epitopes are covered by our product. The clinical design is a classic 3x3 design. It's an open label study to determine the maximum tolerated dose and the fractionation for DSG3 CAR T. Any patient who has tried at least one medicine and had a recurrence is eligible for the trial. In the trial, we have 3 parts. Part A is dose escalation. We go from a subtherapeutic dose to 2 doses that are commonly used in the treatment of cancer patients to a dose that is higher than that. And after we go through that dosing escalation phase and identify a safe and maximally tolerated dose, we then consolidate from a 4-fraction regimen given within a 1-week period to each patient down to a single infusion in Part B. Part C was originally and is currently designed as an open label extension or expansion, but FDA has asked us to consider putting in a comparison arm in order that Part C could represent part or all of the efficacy data in our label if data suggest that the product is safe and effective. The primary endpoint is at 3 months, the safety, the adverse events, and dose-limiting toxicities. The secondary endpoints, very importantly, will include whether or not there is evidence of target engagement with the proximate cause of the disease, the DSG3 antibody. Shown on this slide, the timeline across the top is where I'd like to direct your attention, and you can see that day 8 acute safety data is the primary safety assessment that we are going to be talking about with investors on a dose cohort basis. The reason is that within the first 8 days, the 2 primary adverse events with CAR T therapy, the CRS and neurotoxicity, are both going to occur in that window about 95% of the time. And as a result, if we pass through that window with a clean safety profile, we know that we now have one challenge left, which is to identify target engagement. target engagement is going to be best identified by a reduction in the desmoglein-3 antibody titer, which is measured by a CLIA-approved assay. Because the half-life of these antibodies is 3 or 4 weeks, it'll take about 3 months minimally to be able to definitively understand whether or not we have eliminated the B cells that are making the antibodies, because the antibodies that were made before our therapy was administered will persist for these few months after therapy is administered. We think the risks of safety are very manageable because we only kill 1% of the B cell population, the normal B cell population. The other 99% we don't touch, and there is no tumor, unlike the patients who get CAR T therapy for cancer. So we have many logs fewer cells killed than in cancer patients. That seems to be the best predictor of whether or not you'll see CRS, and particularly whether you would see severe CRS. So patients are treated on an outpatient basis, or if preferred by the investigator, it could be inpatient in this trial. Let me shift very quickly to the Musk Myasthenia Gravis program. That program was designed and is modeled after the DSG3 CAR T program, similar to Pemphigus vulgaris and similar to our discussion from a few slides back. This is a disease where the autoantibody titers drop after rituximab therapy, where the B cell is necessary and sufficient, and where elimination of the B cell and the antibodies that it makes should result in a deep and durable, perhaps a curative response. The structure of the Musk protein is quite similar by chance to the structure of the DSG3 protein. You can see on the right the portion of patients in the myasthenia gravis universe that have the Musk form of myasthenia gravis, and notably, it is typically a more severe form of disease. The acetylcholinesterase inhibitors do not work in these patients. Soliris doesn't even work in these patients. You have very limited treatment options, and the onset of disease tends to be in females at a young age. And so a lifetime of annual or much more frequent administration of therapy is what they're looking at with a revolving door disease state. This is just a little bit of data, a sample of data. We've presented at multiple meetings the in vitro and the in vivo evidence that our Musk CAR T product addresses and lyses or kills the target, no matter which of these epitopes is triggering the disease. We've also done what we believe is the most comprehensive look at the toxicity profile that we can expect in patients. And similar to the DSG3 program, there is no evidence of any off-target toxicity, which shouldn't be surprising because the targeting domain that we use is simply the human antigen. This is in vivo data that we've presented previously at medical meetings. And what you can see here is that the Musk CAR eliminates all of the target cells, but the non-transduced and the DSG3 product, EC1 through 3, do not and are not designed to eliminate the Musk CAR. As a positive control, the CART19, which kills all B cells, does also eliminate the Musk cells. In terms of manufacturing, we've taken an approach that is to minimize risk in the initial lead program. We've partnered with Penn, and we are using exactly the same manufacturing standard operating procedures as were filed in the original IND for Kymriah by the University of Pennsylvania. Penn and its manufacturing facilities and experts are providing us with partnership that allows for production of our product for the lead program. That'll be followed by the partnership that we've recently formed with WuXi in order to support our Musk Myasthenia Gravis program, which we expect, as I said, to enter the clinic after IND filing later this year. And then finally, when we have sufficient evidence from our clinical trials, we intend to build out our own full-scale manufacturing capabilities in-house. So in summary, this is a year that's chock full of catalysts in the clinical program. We expect to be able to report out in the next few months, the acute safety data from the low-dose cohort, followed by continued follow-up, including target engagement in that low-dose subtherapeutic cohort, and any subsequent completed cohorts that we might be in a position to report out, we would do that as well. The Musk IND filing is also expected to occur in the second half of this year. I appreciate your time and your attention. Thank you.